Hello, in this video we will discuss about the interferons, which that is the topic of related to the immune system. So the interferon is denoted by the IFNS. So the interferon means the interfere with viral replication. So it means when viral replicate into the cell of the human body or some animal, so it will be replicate, and inhibition from the replication of viral replication inhibition or something which that is related and interfere with the viral replication is known as the interferon. So that is why its name is the inter. So it is basically produced by human body cells in response to invasion by several pathogens, for example antigen, virus, bacteria and parasites. So on the other hand, one more thing is important is the tumor and cancer cells, which that is defected cell is also produced interferon. So that means it is basically important to know that we will discuss in this diagram. So type of interferon is two, type 1 and type 2. So the interferons type 1 is contain interferon alpha and interferon beta, while the type 2 interferon is the interferon gamma. But what is the interferon? So let's interferon is important for giving a signal to the cell. So it is also known as the signaling molecule, but it's produced by human body cell in response to invasion by several pathogens. So it's basically related to the pathogen interfere with the viral replication inside of the cell. So the identification basically about that is a smaller molecule. So the identical structure and function. So there both type is the identical, and interferon alpha secreted by virally infected leukocytes. So the virally infected leukocytes is secrete the interferon alpha, for example monocyte, macrophage and some leukocyte. So the interferon beta is secreted by fibroblast. So the fibroblasts are present in the tissue, which that is produce the interferon alpha and interferon beta. So this is the basically fibroblasts will be produce and bind with the natural killer cell to ultimately the uninfected cell with viral replication will be killed, and to produce the interferon alpha and interferon beta, and that's interferon type 2 will be produced to prevent the replication inside of the cell. So let's talk about here. So the role of interferon in an antiviral defense. So here is the cell which that is the infected. So that means it is infected cell is produce the interferon and recruit the T cell and natural killer cell. It means the signaling molecule which that recruit the leukocytes. So when leukocyte will be enter, and that means its natural killer cell will be killed, or T cytotoxic T cell will be killed to produce to give apoptosis, while that is the viral genome are present inside. So it is the target cell. So the target cell is here, which that is infected by the virus, while on the other hand here is the uninfected cell which that is signaling by the type 1 interferon alpha and interferon beta will be released from the target cell to basically healthy cell which that is uninfected to give the signaling. So it means the interferon is much more throughout the plasma membrane of all cell. So it means the antiviral protein which that is produced by the by the cascade of the mechanism to signaling of the DNA transcription to produce translation to produce antiviral protein. So this inactive protein is basically activate and inhibit viral protein synthesis and decrease viral nucleic acid. So it means the inhibition of the infection of the virus. So that's antigen inhibition mechanism. While here is the type 2 interferon, interferon gamma, is the produced by activated T lymphocyte and natural killer cell. So it means the type 2 is produced from the cell, and they basically stimulate the activity of macrophage. So it stimulates the activity of the innate immune system and also stimulate the adaptive immune system by the proliferation of T helper cell and cytotoxic T cell. So here is the dendritic cell, which that is contain toll-like receptor, and it is the basically recognize the antigen and give the signal basically. So the dendritic cell will represent the antigen, and that T cell will be recognized, and it contains CD4 receptor and TCR T cell receptor, which that produce the interferon gamma, which that bind with the macrophage and activate the macrophage to engulf it. So it means here is the basically also proliferation of the B cell by the recognition of the antigen, and this B cell is proliferate by the T helper cell receptor to bind with the B cell after the recognition of the antigen from the dendritic cell to that B memory cell proliferation and plasma cell, which that is the after the recovery of the pathogenicity or basically is the infection and the next infection precaution and next infection alertness from the B memory cell to produce antibodies, immunoglobulin. While here the plasma cell on the basis of the time when infection occur is at the time of infection it's produce the antibodies, which that is cover the antigen and give the signal to the macrophage to engulf it. So remember about that. So why basically after the recognition of the antigen from the dendritic cell to produce the interferon gamma, and that interferon gamma will bind with the macrophage activator, so that is innate immune system. So the macrophage is also engulf the antibody containing complex to engulf, and that means the basically endocytosis. So lysozyme enzyme are present inside of the macrophage. So on the other hand, cytotoxic T cell which also basically kill the infected cancerous cell. So thanks for watching.